Hi, my name is Detlef Latzik. I work in the Department of Soil System Science at the Helmholtz Center for Environmental Research. Our name represents our scientific interest in a deep understanding of the diverse interactions in the habitat formed by a soil. Plants interact via their roots with the microorganisms, fungi, algae, and soil animals, which live in a soil in extreme diversity. Where life exists, nutrients and energy are needed. The oxygen diffusing into the soil is urgently needed by the communities living there to metabolize carbon-containing substrates. CO2 is formed as a result of this soil respiration. From local observation, we know that the respiration rate of the soil ecosystem depends on biotic and abiotic factors, such as the availability of easily convertible carbon sources, root penetration, temperature, the soil water content, soil aeration, and so on. If we know the amount of CO2 that is produced in the soil, we learn something about the living conditions in the soil. That is about its fertility and the health of the soil ecosystem. With this knowledge, new opportunities arise to better understand and specifically influence soil processes on larger scales. But how can we measure CO2 in the soil? The soil pores between the grains are tiny, and the soil properties vary strongly from one place to the next, even in a single field. And because properties such as temperature, water content, the nutrient availability vary in time, so will the local CO2 concentration of the soil. We therefore need to measure the average CO2 concentration of the soil over a relatively large area so that we sample a very large number of soil pores. In our article in Vadose Zone Journal, we present a CO2 sensor that consists of a flexible tube made of a material that allows gas to diffuse through it. We can bury this tube in the soil, purge it with air, seal it for a short time, and measure the pressure change during that time. By comparing this pressure change to that of a reference system for which we use a defined CO2 concentration inside of a second tube, we can derive the CO2 concentration in the soil averaged over the entire tube length. We can carry out these measurements at the end of the tube that stick out of the soil. We tested the sensor in several large soil containers planted with barley. We installed tubes at different depths and measured CO2 concentrations during the entire growth cycle of the barley every 2.2 minutes. Our container experiments were carried out under the same external conditions. Nevertheless, the growth rate of the barley varied between containers. It was astonishing for us to see how clearly the dynamics of the CO2 signal correlated with these differences. This means we were able to demonstrate that the measurement signal of this novel CO2 sensor indeed reflects the average soil CO2 concentration, and the signal is not dominated by local variations. By carefully choosing the length of the tube, we can use the CO2 readings to learn about carbon turnover and gas exchange in a soil area of any desirable size. Our open access article in Vadose Zone Journal is freely available. Megan Silver discussed this new technology in a featured article in the October 2019 CSA News. Thank you for your attention. My co-authors and I look forward to talking to you